Hi everyone, my name is Angel de Alba, and today we're going to talk about this amazing bacterium called Escherichia coli, or E. coli as it is also known. Let's start off with the history of this bacterium. Escherichia coli was discovered in 1885 by the German-Austrian microbiologist and pediatrician Theodor Escherick, while he was conducting examinations on neonates' meconium and feces of breastfed infants. He first called this rod-shaped bacterium Bacterium coli commune, but it was renamed as Escherichia coli after its discovery in 1919. E. coli's rise among the scientific community was due in large part to the fact that it was easy to find and work with, which established it as the bacterial model organism of choice. In fact, the most basic aspects of life, such as the genetic code, transcription, translation, and replication, were first worked out in E. coli. Right here, we have a picture of Escherich's drawings of the bacteria he observed under the microscope while he was conducting his examinations of baby's stool. Let's now talk about its classification. E. coli is classified as a bacterium because it is unicellular. It belongs to the phylum Proteobacteria as it is a gram-negative microorganism, meaning that it has a thin cell wall composed of a few layers of peptidoglycan and additional structures surrounded by an outer membrane. As you can see in this picture, we have a plasma membrane, we have a peptidoglycan cell wall, and we have the outer membrane. This bacterium is also part of the gamma proteobacteria class as it is facultatively anaerobic, meaning that it can survive with and without oxygen. Furthermore, it belongs to the Enterobacteriales order because it is gram-negative, facultatively anaerobic, and also rod-shaped. These three characteristics, along with motility via flagella and other characteristics, put this bacterium into the Enterobacteriaceae family. Its genus, Escherichia, is named after Theodor Escherich, who, as we know, was the person who discovered this genus. Let's now look at its structure. As most bacteria, E. coli has DNA. However, unlike eukaryotes, E. coli does not have a nucleus to enclose DNA. Instead, DNA is located in a region called the nucleoid, which is usually located in the central region of the cell. It has to be noted that E. coli has a single circular molecule of DNA that is condensed. Ribosomes can also be found in this bacterium, as well as a cytoplasm. Because E. coli is a bacterium, membrane-bound organelles are not found in it. Moreover, this bacterium has a cell envelope composed of the inner membrane, which is a plasma membrane, a cell wall, and an outer membrane. This envelope defines cell shape and allows the cell to withstand large mechanical loads, such as torque or pressure. The inner and outer membranes are separated by a region called the periplasmic space, where a rigid peptidoglycan cell wall is found. The inner, or plasma membrane, is a phospholipid bilayer that is selectively permeable, while the outer membrane is composed of phospholipids in the inside leaflet and lipopolysaccharides in the outside leaflet. Recent reports have actually shown that the stiffness of the cell envelope is largely due to the outer membrane. The outer membrane also contains proteins such as OMP-C and OMPF, which are foreign proteins and which regulate the passage of small solutes into the cell interior. Some strains of E. coli also exhibit a capsule, which is a hydrophilic layer composed of polysaccharides that envelopes the bacteria. A plasmid is also usually present, which is a small circular DNA molecule found in bacteria that replicates independently from the bacterial DNA. Other features of this bacterium include many flagella and fimbriae, which are filamentous surface organelles. Flagella aid with motility of the cell, while fimbriae, which are shorter than flagella, allow the cell to adhere to biotic and abiotic surfaces that lead to the formation of biofilms and colonies of bacteria. Pili, which are filamentous surface organelles as flagella and fimbriae, may also be present to aid with adherence to other cells and surfaces. This is a picture of the cell envelope found in E. coli. As you can see in the picture, we have a semi-permeable plasma membrane, the periplasmic space where the peptidoglycan cell wall is found, and the outer membrane composed of phospholipids in the inside leaflet and lipopolysaccharides in the outside leaflet. So now, how does E. coli reproduce? As all other prokaryotes, E. coli reproduces asexually through a process called binary fission. After DNA replication, which also includes the replication of the plasmid, and which occurs in a similar way as in eukaryotes, each of the two copies of genetic material move to opposite ends of E. coli and attach to the plasma membrane. Then, E. coli continues to grow and elongates, eventually pinching itself inward at its equator and producing two clones. Under suitable conditions, binary fission can occur every 20 minutes. E. coli also undergoes conjugation, a process by which one coli transfers genetic material to another coli. This happens between an E. coli that has a plasmid, which is a donor, and another without a plasmid, which acts as the recipient. Once the plasmid in the donor is replicated, the bacterium releases an enzyme that has a mating signal, which indicates that it is ready to undergo conjugation. The recipient bacterium is attracted to the donor, which uses a sex pilus to connect both bacteria. The donor transfers the plasmid, contributing to the alteration of E. coli's genetic makeup. Now, you might be wondering, does E. coli cause any diseases? Well, E. coli is mostly a harmless bacteria found in our intestine. However, there are some strains that cause disease, such as E. coli O157H7, which produces a toxin called Shiga. This toxin damages the lining of your intestine and as a consequence causes symptoms like abdominal cramps, vomiting, diarrhea, which might also include bloody diarrhea and sometimes fever. This strain of E. coli is the leading cause of hemolytic uremic syndrome or kidney failure. So, how do you get infected by E. coli O157? Well, people who consume meat that wasn't cooked thoroughly, especially ground beef, might get infected by E. coli O157. This is because when meat is processed, some of the bacteria found in the animal's intestines make their way into the meat. Similarly, consuming unpasteurized milk, which is milk that was not subjected to heat in order to kill pathogenic microorganisms, can also infect people with this strain of E. coli. Furthermore, consuming fresh vegetables and fruits that have been washed with water can infect people with the bacterium since the water can be contaminated with the bacteria. Other foods, such as yogurt and cheese, made from raw milk can also infect people, as well as unpasteurized juices. Animals, such as those seen at fairs and zoos, can have the bacteria and thus infect people who touch or hang around nearby animals. People who are infected with the bacteria usually experience symptoms within a range of 3 to 4 days. So, how do you treat an infection by E. coli O157? Well, most of the time, the infection goes away on its own within 5 to 10 days. Now, diseases are typically treated with antibiotics. However, this is not the case. Why? Because antibiotics might increase the production of Shiga toxin and worsen symptoms. Instead, patients are encouraged to have enough rest, consume plenty of fluids to replenish everything lost through vomiting and diarrhea, and also practice good hygiene habits. People who develop kidney failure may require hospitalization, where blood transfusions and or kidney dialysis might be done. It is also important to cook meat well, enough to kill any bacteria found in it, consume pasteurized products, and thoroughly wash fruits and vegetables with clean, uncontaminated water before consuming them. Let's now take a look at other cool facts about E. coli. Because of its hardiness, versatility, and ease of manipulation, E. coli is the most studied and best known organism in the world. It has actually been used for biotechnological purposes, such as making human insulin. Scientists achieved this by extracting a small sequence of the gene that codes for insulin in human cells and inserting it into E. coli via a plasmid. Once a human gene is inserted into the bacteria via the plasmid, the bacteria will copy their own DNA along with the plasmid and will divide, passing on the plasmid with the insulin gene to new bacteria so that division occurs again. Scientists then extract the insulin from the bacteria and purify it for human use. E. coli is also resistant to several antibiotics. It is actually believed that this antibiotic resistance is largely due to the outer membrane structure, which does not allow E. coli to be killed by antibiotics. And this picture right here shows how scientists use E. coli to make human insulin. Pretty cool, huh? This is a picture that shows the resistance percentage of E. coli to different antibiotics. As you can see, E. coli has a resistance percentage of 100% to antibiotics like ampicillin and cephalothin. Similarly, E. coli has 94% resistance to streptomycin and 92% resistance to tetracycline. This antibiotic resistance is very common in different strains of the bacteria. E. coli's growing resistance to antibiotics is of great concern in the scientific community, as this bacterium is the most common pathogen we as humans are exposed to. Efforts are being made to stop the growth and spread of antibiotic resistance among E. coli strains to avoid further consequences. Alright everybody, that's it for today. Thank you so much for watching this presentation. I hope you enjoyed it, and I hope you learned a lot about this amazing bacteria.